Uranus. Where did it come from, and how did it get where it is? No, I'm not talking about the planet. I'm talking about your... Biologists continue to debate the origins of the anus, a structure which resulted in a highly successful lineage of organisms known as the Bilateria nephrozoa, or animals with a symmetrical body plan and 2 digestive openings. We may take for granted that we have a through gut, allowing for one-way flow for digestion. But organisms possess a wide array of specialized structures for the breakdown of food, absorption of nutrients, and defecation of excrement, all controlled during development by a multitude of genes and enzymes. In fact, some animals only have one alimentary opening, requiring them to eat and excrete from the same place. Understanding more about the formation and development of the different parts of the gut can help us understand how to identify and treat different types of cancers, like colorectal cancer. So, let's get to the bottom of it. I'm Professor Sunny, and thanks for joining me as I start asking questions. Organisms require nutrients as an energy source to maintain their body structures and to reproduce, or to simply go about the chore of living their lives. To do this, the life on Earth has formed many different body plans to take advantage of the plethora of environmental niches available. In fact, it's safe to say that there's hardly an aseptic location that we've yet found on Earth, except perhaps the hot, salty, and highly acidic waters of the Dalal geothermal field. Plants often use roots to absorb nutrients, chlorophyll to capture light, and pores to bring in carbon. Bacteria receive their nutrients by absorption or phagocytosis. But as for the animal kingdom, not all of its members feature bilateral symmetry with a through gut. For example, sponges are filter feeders with pores that open into a central absorptive chamber called the spongiocele. In contrast, jellyfish have to expel their food waste back out of their mouths before being able to consume more. Interestingly, the jellyfish's gonads are located within the walls of their stomach. In contrast, bilateria, like ourselves, are fortunate enough to have a generally one-way digestive tract. This gives us the adaptive benefit of being able to continue to consume food without having to wait to excrete our previous meal back out of the same orifice it entered. In most bilateria with a through gut, it's further specialized into 3 regions: a foregut, midgut, and hindgut, each with different functions. The mouth is specialized with teeth and muscle to masticate our food and direct it toward the foregut, which includes the stomach. The stomach further macerates the food bolus to prepare it for chemical and enzymatic catabolism. The midgut is where bile chemically breaks down and frees nutrients, which are then absorbed into the body's circulation. The hindgut, which includes the descending colon, is specialized in liquid reabsorption. But the hindgut does not form from the end of the midgut. Instead, both the urinary bladder and the hindgut begin as pores or invaginations formed from the embryonic cloaca. The hindgut then connects to the midgut to form the anorectal canal and the distal colon. But first, let's take a step back to look at development in general. How is it that one single cell leads to such a complex and multicellular body? The process begins at the first moment the spermatocyte comes into contact with the ovum. At this moment, the point of contact becomes the animal pole, and the opposite side becomes what's called the vegetal pole, creating a spatial and biochemical gradient across the fertilized egg cell. Developmental signals are released in the form of chemical molecules, and as they diffuse across the cell, the concentration of those molecules decreases with distance. Where the concentration is higher, more of the molecules can bind to receptors, creating a strong effect. At the far end, fewer molecules are present, so the effect is weaker, or the tipping point isn't even reached. This principle of the chemical concentration gradient is the fundamental force that drives the differentiation into different cell types and different body plans by activating different genes. The genes that help segment the body into its different regions from head to tail are called homeobox genes, or Hox genes for short. And these Hox genes are conserved across a wide range of species, from snails to humans. When a gene is unchanged across so wide a range of species, that means it's important. For example, one Hox-related gene among many that triggers the formation of the hindgut is called CDX2. If an organism has mutations that inactivate its copies of CDX2, then the embryo is not even viable. That's how important these Hox genes are. Interestingly, a group of researchers have recently released a preprint report on a group of bilateria known as Xenocoelomorpha. These tiny invertebrates feature a blind gut with a single opening without any hindgut. So much like the jellyfish, they expel their excrement from the same place it entered. Interestingly, the researchers extensively showed that the xenocoelomorpha still expressed hindgut Hox marker genes, even without a hindgut to speak of. These hindgut marker genes were present around the caudal or tail end of the animal in a region called the male gonopore, which is a small genital opening from which sperm are released. The researchers thus conclude that the bilaterian anus evolved from the male gonopore. So basically, we have the male sex to thank for our. These conclusions also aim to reposition xenocoelomorpha to a more important role in the bilateria lineage. As a preprint article, this is a scientific manuscript that's not yet been peer-reviewed. Scientists will often submit preprint manuscripts to garner feedback from the broader scientific community with the aim of improving their study before submission to a scientific journal. Whether the molecular and genetic evidence presented in this research will stand up to scrutiny is yet to be seen. Alternative explanations could be that evolutionarily earlier xenocoelomorpha did possess a through gut, which in later evolutionary time closed, and the male gonopore was never involved. The hindgut genes observed around the gonopore could have simply been used as a convenience in forming these structures during development. But the authors argue that the developmental presence of the mammalian cloaca, which goes on to form the urogenital system but not the gonads, is morphologically similar and constitutes evidence of this connection. So, what do you think? Is this snapshot of the morphology of different species enough to convince you about the evolution of the through gut? In the end, there's still probably not enough information to say definitively, and the scientific debate is sure to continue regarding the origins of Uranus. So, if you're interested in this sort of thing, be sure to keep asking questions. Interestingly, these Hox-related genes like CDX2 may play a future role in cancer screening. Continued expression of CDX2 throughout life is important for maintaining the specific functions and traits of the colon. Because of this, CDX2 has been studied in the scientific literature and is associated with an interesting role in patient mortality with colorectal cancer. Often, when a colon cell loses CDX2 expression, a patient will have a poorer prognosis. Conceptually, that cell has an increased risk of spreading or metastasizing throughout the body because, through mutation, it has lost its affiliation with that region of the body. However, CDX2 is not yet used as a routine biomarker, as more definitive work's needed to ensure its accuracy and its prognostic value. If you are concerned about colorectal cancer, I suggest talking to a licensed medical worker. There are a number of genetic, dietary, and lifestyle factors associated with an increased risk of colorectal cancer, and routine screening is now recommended for adults as young as 45 and up to 75 years of age, especially if they present with symptoms such as rectal bleeding, iron deficiency, abdominal pain, or unexplained weight loss. Factors that can increase your risk include a history of polyps or the presence of large polyps, inflammatory bowel disease, cystic fibrosis, and a family history of the condition. Long-term cigarette smoking is associated with an increased incidence and increased mortality rate, as well as the consumption of red meat and processed meats. Heavy alcohol consumption, obesity, and low vitamin D levels round out the risks further. Reduced risks are associated with regular physical activity, diets high in fruits and vegetables, and dietary nutrient supplementation, as well as the cessation of smoking. A number of genetic or familial syndromes carry an increased risk of colorectal cancer and can include the involvement of a range of well-studied genes involved in polyposis, Lynch syndrome, and Putz-Jaeger syndrome, to name a few. I am not a medical doctor, and nothing I say should be construed as medical advice. So if you have questions about your specific health, then please talk with your trusted healthcare provider, and not me. Thanks for watching.